Bismillahirrahmanirrahim. Assalamu alaikum. The topic for today's lecture is the prevention of blood clotting in the normal vascular system, that is the intravascular anticoagulants. The role of the intravascular anticoagulants. Before our today's discussion, I would like to summarize the previous lecture also. We have discussed completely the extrinsic-intrinsic pathway for coagulation and their common pathway at the step where the factor 10 is activated. In this figure, I want to just summarize the previous lecture in which the extrinsic pathway is shown in pink color, while intrinsic pathway in which the color is blue and the common pathway shown by the grey color, and also the other clotting factors. Name of the other clotting factors is also written here. So this a single picture is going to show all these pathways separately and also its common pathway. So if you remember just the single picture, this one, you can show all the clotting mechanism completely. Now coming to the today's lecture. In our body, either the coagulation process will take place or not. This thing is decided by the fact that how the anticoagulants they are activated or the procoagulants they are activated. Some of the agents in the blood they are going to promote the process of the coagulation, and some they act against that one, that is they stop the process of the coagulation. The balance between these two types of the agents that will decide whether the coagulation process will take place or not. Regarding the intravascular anticoagulants, that is the prevention of blood clotting in normal vascular system. This is a very important topic, but on the other hand, more important thing is that once the coagulation process takes place at local site, how the and all the clotting factors they are present in the blood, how the other sites of our body that is prevented from the excessive coagulation. So clotting must take place at the injured site, not at other or in the whole of the body. How this mechanism take place so that only clotting will take place at the site of the injury. The other sites, healthy sites, they will be protected. And secondly, how this process is going to be stopped. Otherwise, it will go on. So what's its check? This is the whole thing that we will discuss in today's lecture. Normally, the endothelial surface factors, that is the most important thing, is the smoothness of the wall and the glycocalyx thrombomodulin, which is going to bind the thrombin, protein C, which is going to inhibit the factor 5 and 8. The second point is antithrombin action of the fibrin and antithrombin 3. Thirdly, heparin release by basophils and mast cells. And lastly, the role of the plasmin, which digests fibrin fibers and other clotting factors. Now, I would like to discuss all these one by one. Regarding the endothelial surface factors, the smoothness of the endothelial surface prevents the contact activation of the intrinsic clotting system. A layer of glycocalyx on the endothelium, which repels clotting factors and platelets, thereby preventing activation of clotting. And thirdly, the thrombomodeling binds thrombin, which activates the protein C. Actually, this act as an anticoagulant by inactivating activated factors 5th and the 8th. So it inactivates these factors and act as anticoagulant. Secondly, antithrombin action of the fibrin and antithrombin 3. Among the most important anticoagulants in the blood are those that remove thrombin from the blood. The most powerful of these are, number 1 is the fibrin fibers that are formed during the process of clotting, and secondly, antithrombin 3. While a clot is forming, almost about 85 to 90% of the thrombin formed from the prothrombin becomes absorbed to the fibrin fibers as they develop. This helps prevent the spread of thrombin into the remaining blood and therefore prevents excessive spread of the clot. The thrombin that does not absorb to the fibrin fibers soon combines with antithrombin 3, which further blocks the effect of thrombin on the fibrinogen and then also inactivates the thrombin itself during the next 12 to 20 hours. Regarding heparin, which is another powerful anticoagulant, but its concentration actually in the blood is normally very low. So only under special physiological condition it has significant anticoagulant effect. By itself it has little or no anticoagulant properties actually. But when it combines, I would like to show you this diagram, this animation, so that actually the heparin is itself has no role as anticoagulant properties, but when it combines with antithrombin 3, the effectiveness of antithrombin 3 for removing thrombin increases by 100-fold to 1,000-fold, and thus it acts as an anticoagulant. Therefore, in the presence of excess heparin, removal of free thrombin from the circulating blood by antithrombin 3 is almost instantaneous. So it is going to speed up the process. The complex of heparin and antithrombin 3 removes several other activated coagulation factors in addition to thrombin. Now comes the role of the plasmin. Mainly, the role is the lysis of the blood clots. The plasma proteins contain plasminogen, which is the inactive form, and its active form is the plasmin. Plasmin digests fibrin fibers and some other protein coagulants such as fibrinogen, factor 5, factor 8, prothrombin and factor 12. Therefore, whenever plasmin is formed, it can cause lysis of a clot by destroying many of the clotting factors. So, sometimes even causing hypocoagulability of the blood also. When a clot is formed, a large amount of plasminogen is trapped in the clot. So, how the plasminogen is formed and how the plasminogen is converted into plasmin and how it causes the lysis of clot. Actually, when a clot is formed, a large amount of plasminogen is trapped in the clot. This will not become plasmin or cause lysis of the clot until it is activated. That is decided. Plasminogen is in activated form while its activated form is plasmin. The injured tissues and vascular endothelium very slowly release a powerful activator. That is very important thing. Tissue plasminogen activator. That a few days later, after the clot has stopped the bleeding, eventually converts the plasminogen to plasmin. So, plasminogen is kept inside the clot unless and until the TPA eventually converts plasminogen to plasmin, and after some days when the bleeding stops, then this conversion takes place, and in turn it removes the remaining unnecessary blood clot. A very important thing which should be noted here also, that many small blood vessels in which blood flow has been blocked by clots are reopened by this mechanism, and this is a very advantageous step of this conversion of plasminogen to plasmin after some days because of the activation of the TPA. It is very helpful for other sites also. Thus, an especially important function of plasmin system is to remove minute clots from millions of tiny peripheral vessels that eventually would become occluded were there no way to clear them. And this is the thing which I was talking previously, that how this process stops and also helpful for the other sites, so that on the healthy sides, number one thing, the coagulation should not take place, as all these factors, they are attracted to the injured site and they are utilized there. They are not present, they don't remain in the blood to act on healthy sites. Number one thing, and once this plasminogen and plasmin that is activated, the lysis of the clots take place on that place, on the already injured site where the newly formed clot is there, and also help for the other sites for removal for the minute types of the clots. This is a very nice figure to show what is the action of the plasminogen, which is the inactive form, and it is converted into plasmin. To consider this or to understand this figure completely, you must know that blue arrows is showing the stimulation, while the red arrows, they are meant for the inhibitory effect. Almost this is the whole of the summary, which is started, the process starting from the thrombomodeling thrombin complex. Then protein C is converted into activated protein C, and so on. And you can see that the plasminogen is converted into plasmin, and then the role of the DPA, so that the lysis of fibrin clot, newly formed clot, that takes place. So this is all for the today's lecture. Okay, thank you and Allah Hafiz.